Thank you. Excuse me. It's a great honor to be here. So thank you for inviting me. And I'm looking forward to the discussion after the talk. I'm a reproductive geneticist, and I'm interested in the process of making eggs and sperm, specifically how the processes are controlled, the errors that occur, and the differences between males and females. One of the most interesting things about human reproduction is that we, women, make a lot of mistakes. I began my research training in a lab that was trying to determine how common chromosomally abnormal pregnancies are. And we knew that the risk of a child with Down syndrome is much greater for women in their 30s and their 40s. And Down syndrome results from an extra chromosome 21, which is the smallest chromosome in our genome. Because there's less genetic information on this chromosome, babies with an extra chromosome 21 survive. What about other chromosomes? To find out, we studied human miscarriages. And what our laboratory and several others found is that mistakes in other chromosomes are also increased in older women. But the effect's very interesting. This graph shows the frequency of these errors, which we call trisomy, an extra chromosome, plotted against maternal age. So you can see that the risk increases suddenly and very precipitously after the age of 30. So much so that we estimate that by the time a woman is in her 40s, about half the eggs she's ovulating must be chromosomally abnormal. But look a little more closely at this curve. There's also an increase among the youngest mothers. Hormones control the growth and the maturation of the eggs we ovulate. But we also know that menstrual cycles are erratic in the years right after puberty and as menopause approaches. Because hormones control the growth and maturation of the egg, this made me wonder if you could argue that the beginning of our reproductive lifespan and the end of our reproductive lifespan, at those times, the ovary doesn't provide an ideal environment for making a perfect egg. In genetics, the mouse is an important tool to study complex genetic processes. By comparison with humans, female mice don't make a lot of mistakes in making eggs, and there isn't a strong effect of age. But I wondered if studying mouse eggs might not provide us a way to assess the impact of subtle changes in hormones and the signals they're sending. And imagine my surprise when our data in mice suggested that subtle hormone changes, in fact, increased the number of eggs with chromosome errors. But before we could announce our findings, we ran into a problem. Suddenly, eggs from normal females had a lot of errors. Right before the egg is ovulated, it eliminates half of its chromosomes. And this is essential because we need a sperm with half the number of chromosomes and an egg with half the number of chromosomes to create an embryo with the right number of chromosomes. After many months of trying to understand the sudden change in eggs from normal control females, we determined that there'd been an accident in the mouse room, a mistake in the cage washing procedures. A temporary worker used the wrong detergent on the cage washer, the floor detergent, and this damaged the plastic cages and water bottles, and they began to leach bisphenol A, or BPA. What we're seeing was the result of BPA exposure. An example is the picture on the left. Those little red blobs are the chromosomes, which should be nice and tightly aligned, and instead they're splattered all over the place. And what happened as a result of this disorganization is an increase in chromosomally abnormal eggs. So as I'm sure all of you know, BPA is one of those endocrine disrupting chemicals that Pete Myers talked about a few weeks ago for this group, a man-made chemical that can mimic or interfere with our body's hormones. When we published our findings 20 years ago, they received a lot of media coverage because of the human implications. This chemical that we're all exposed to on a daily basis, if humans behave like mice, exposure to this chemical would cause an increase in miscarriages or babies born with birth defects. And at the time, all the baby bottles in the United States, I'm not sure about Japan, were made of polycarbonate plastic, made with BPA. So this was obviously of concern to me, and since I'm interested in the process of making eggs, let's back up and think about how an egg is made, because it's actually fascinating. The process starts during fetal development, and it takes decades. As the ovary is developing in the female fetus, all of the eggs that the adult female will ovulate are starting their development. Some really important events take place during this fetal process, and then the egg enters a sort of suspended animation, like Sleeping Beauty, and it remains suspended for a very long period of time. But in the perinatal period, just before birth in humans, the egg becomes surrounded by the follicles. And when the female becomes an adult and the hormone signals are appropriate, every cycle, a number of these follicles will start growth, and hopefully the cycle will culminate with the ovulation of a chromosomally normal egg. So our initial accidental exposure to BPA was affecting this adult stage and affecting those one or several eggs that were completing their growth and being ready to ovulate. We now know that exposure to BPA and other bisphenols and some of the other endocrine disrupting chemicals can affect all three stages of egg development. And we focused on this earlier stage, the fetal stage of development, because, as I said, some very important processes take place here. We didn't expect to see any effect because, as far as we knew, hormone control was not what drove this fetal portion of the process of making an egg. To our surprise, we saw some subtle but very important changes to the eggs from exposed females. And when we let these females who were exposed in utero, remember, when we let them grow into mature adults, we found a dramatic increase in chromosomally abnormal eggs. Thus, we have two different developmental stages during which exposure to BPA can adversely affect the genetic quality of the eggs produced. This fetal stage is very important for two different reasons. First, unlike our first inadvertent exposure to BPA, where only those eggs that were in the final stages of growth and ready for ovulation were affected, at this point, all of the eggs the female will develop are starting their process of development. And so exposure here can impact the entire cohort of eggs that female will produce. In addition, this is what we call a grand maternal effect, although I think it's easier to think of this as a single exposure that hits 3 generations simultaneously. We're exposing the pregnant female, and BPA can cross the placenta and affect her developing fetus. And if that fetus is female, it can impact the eggs in that developing ovary, which will give rise to her grandchildren. So what's important here is that a single exposure can affect 3 generations at once. So this is a very serious effect in my mind, and although it didn't generate the same media attention as our first inadvertent exposure to BPA, my scientific colleagues were stunned. So in addition to our studies in mice, they tested the effect in rhesus monkey females and in a small roundworm, a nematode that geneticists who study egg development love to use because these worms are transparent, so you can actually see the eggs developing inside of them. Remarkably, the same effect of BPA exposure on the earliest stages of egg development were evident in these 3 very different species. Obviously, this raises huge concerns for humans, but it's important to remember that BPA is but one of a large group of endocrine disrupting chemicals. And as more adverse effects were reported for BPA, replacement bisphenols came on the market. And we tested some of them, but could by no means test all of them, and found similar effects on the early stages of egg development. We also found that some members of another class of plasticizing chemicals, the phthalates, would induce the same effects. But there are a whole host of other endocrine disrupting chemicals, pesticides and insecticides, the perfluorinated chemicals, the persistent organic pollutants like DDT and PCBs, flame retardant chemicals, and some of the heavy metals like lead and mercury. Obviously, we can't test all of these. And although I focus on reproduction, it's important to recognize that our endocrine system does far more than make sexual function possible. It controls our metabolism, our growth, our sleep, and our behavior. So these endocrine disrupting chemicals are like radiostatic. They interfere with the very complex signals that control our body and make it harder for our hormones, our body's hormones, to do what they're supposed to do. Additionally, we're most vulnerable to the effects of these endocrine disrupting chemicals during fetal development when our bodies are forming. This quote is from an editorial published 7 years ago in a major medical journal, and it summarizes how I feel about these chemicals. They are a public health problem, but more importantly, they're a planetary health problem. Experimental studies in mice and other animals have suggested that endocrine disrupting chemicals and our exposure to them may contribute to human health problems that have become epidemic, like infertility, obesity and diabetes, autism and heart disease, and even cancers, some cancers. But let's return to reproduction because, as we all know, it takes more than an egg to make a baby. So do these exposure to these chemicals affect the male? My colleague Shauna Swan has been arguing that human sperm counts have declined rapidly during the past several decades, and that this is likely due to our exposure to endocrine disrupting chemicals. Shauna published this book in 2020, and it's been extensively covered in the popular press, including this very fun and accessible cartoon video on YouTube. So let's consider the process of making sperm. It's very different from making an egg. During fetal development, a testis is formed, but we're actually years away from the process of making sperm. Shortly after birth, very special cells, the stem cells that allow the process of spermatogenesis to be continuous, making millions and millions of sperm. So shortly after birth, this specialized set of cells are formed. But the actual process of making sperm doesn't begin until sexual maturation and the production of hormone signals that are essential to controlling the process. Surprisingly, although there's no active sperm production during these two early time points, they're vulnerable to exposure effects. We focused on this neonatal time point and found a very interesting effect. Exposure to bisphenol A and to some of these replacement bisphenols and to phthalates affects the stem cells of the testis. When the male becomes sexually mature and the descendants of these stem cells start making sperm, we saw an increase after exposure in chromosome errors. And that's what you're seeing in this picture on the left, is you're seeing a cell that's making errors in its chromosome behavior. Now, this is a very interesting difference between men and women and between the sexes in mice as well. Unlike the female, where errors that lead to chromosomally abnormal eggs and abnormal embryos, in males, cells that are making mistakes are very effectively eliminated. If enough errors are made, fewer sperm will result. Thus, our findings from studies in mice directly support Shauna Swan's argument that exposures to these types of chemicals may be causing a reduction in sperm production in human men. My colleague at Washington State University, Mike Skinner, has reported some very interesting findings looking at the earliest stage, fetal development, and exposures that take place at this time. Mike Skinner has found that some exposure effects are transgenerational. And by this, he means that the exposure may not only be evident in the exposed individuals, but in his sons, grandsons, and even subsequent generations, great-grandsons. Typically, the way these experiments are done is by exposing during fetal development, not in humans, as I'm illustrating here, but by exposing during fetal development and assessing the effects of exposure when that individual becomes an adult, and then assessing effects in his sons, grandsons, and even great-grandsons. So when we presented our data on the effects of exposure to the testis of newborn mice, our colleagues always asked, is this a transgenerational effect? And a very talented student in my lab, Tegan Horan, decided to take on this question. But in addition to asking whether the effects of exposure were evident in subsequent unexposed generations, Tegan decided to ask a more real-world question. What happened if you had an exposed individual and then the sons and the grandsons were also exposed? In the case of humans, this is more what's happening in our world. In fact, you could argue that the exposure is increasing with subsequent generations. And in addition to finding that the effects of exposure were transgenerational, that's the exposure effects were seen in subsequent generations in grandsons and even great-grandsons, Tegan found with successive generations of exposure, the effects worsened. And in the third generation, a number of males had testes that were not capable of making sperm. In the course of other experiments Tegan was doing, we again began to see signs of contamination. This was almost 20 years after our first exposure in the female mouse. And what we were seeing is that normal control males began to exhibit signs of exposure. Again, the problem proved to be damaged cages, although in this case, it was much milder and gradual than the first exposure. And this time our animals were being exposed to BPS and perhaps a little BPA. Tegan, the clever student that she was, decided to use this second exposure accident to ask an important question. What would happen if we actually could eliminate our exposure to these chemicals? So she asked, could I take these estrogen-exposed males and breed them out for successive generations and ask if their sons, grandsons, and great-grandsons were not exposed, if the effects, how long the effects of exposure lingered. What Tegan found is that the sons showed the same effects of exposure as the fathers. The grandsons were getting close to normal control levels, and the great-grandsons were actually back to normal. So the answer was the effect of exposure disappeared after 3 to 4 generations. So now I want to try to put these findings into a larger context. We know that who we are, what we look like and how we behave is the result of the interaction of our genes and our environment. And so changes in our environment, like an increase in endocrine disrupting chemicals, have the potential to affect us. We also know that we are especially responsive to our environment during fetal development. And stress to the mother can impact the developing fetus, whether it's obesity, starvation, emotional stress, or even some of the tools and processes used in assisted reproductive technology, or ART. So with respect to endocrine disrupting chemicals, this raises several important questions. How much exposure is safe? These are hormone-like chemicals, and because hormones can exert very powerful effects on our cells and our bodies at very, very low levels, some of us would argue that no amount of human exposure should be considered safe. So how exposed are we? And especially during fetal development, since it's such a vulnerable stage. We don't have a terribly good answer to this. It's hard to measure fetal exposure, and especially since that exposure is constantly changing. BPA provides an example of how rapidly human exposure to these chemicals is changing. As more and more data accumulated about the adverse effects of BPA, BPA-free products came on the market. And what these products were essentially BPA being replaced by structurally similar replacement bisphenols. And this is no guarantee that these are safer products. In fact, some of these replacement bisphenols look like they may be even more dangerous. This leads us to the most important questions. Why aren't these chemicals regulated? The short answer is that chemical regulation is mired in the past. When we first started evaluating the safety of chemicals, we were interested in very simple questions. How much exposure is safe? How much is going to kill us? Do these chemicals, or does this chemical behave like a mutagen? Does it alter our DNA? Do these chemicals cause cancer? Because we were interested in these questions, we focused on how much of a chemical exposure would cause these different effects. And the assumption was that there was a linear increase. If a little was harmful, more would be even more harmful, and even more would be more harmful until you finally got to a point where it would kill the animal. In other words, the dose makes the poison. But EDC, endocrine disrupting chemicals, are very different. Hormones are signaling agents. Very small amounts exert very profound effects on our bodies. And chemicals that mimic or interfere with our body's hormones don't play by these simple rules. If a little is bad, more is worse. Sometimes very, very tiny amounts of exposure can have a profound effect, and increasing levels will have a different effect or show no effect. Thus, the old ways of thinking about chemical safety and testing chemicals simply don't work for endocrine disrupting chemicals. So why does this matter? Well, I've shown you that inadvertent exposure to these endocrine disrupting chemicals can disrupt research. And it's not simply that I have profoundly bad luck. It's affecting the research in a lot of different laboratories. It's simply that we've been able to see the contamination as it's sneaked into our laboratory. These exposures can also impact clinical care. For instance, cancer patients who are undergoing treatment. Exposure to some of these estrogen-like chemicals can interfere with some of those treatments. Similarly, for patients undergoing assisted reproductive techniques, some of these hormone-like chemicals can interfere with that process as well. And of course, these chemicals can impact our lives, the lives of our children, and the lives of all the other species that share our planet. So what can you do as a consumer? Well, you can develop what I call chemical awareness, become a savvy consumer, read labels, and avoid some of the common endocrine disrupting chemicals that are added to personal care products and other cleaning products. Treat the plastics in your life with caution. Don't microwave them. Don't put them in the dishwasher. That's an invitation for these chemicals to migrate out. Discard plastics showing signs of wear and tear. Plastics are not forever products, and if they're showing signs of wear and tear, they're leaching chemicals. Most importantly, it's not my research or the research of any other scientist that's going to change things. It's you, the consumer. Ask manufacturers for what you want. Manufacturers listen to consumers, and they will respond. And thank you most of all to my wonderful interpreter, Kat. Thank you.
Thank you.